Good morning, everyone. My name is Liang. I'm a principal scientist in the Discovery Chemistry Department at Genentech. First and foremost, I want to thank Sandia, the organizer, for the opportunity to speak at this symposium. I'm excited to share our research that led to GDC 9545 as a best-in-class molecule. The title of my presentation is The Discovery and Development of Estrogen Receptor Degraders, SERT, for ER-positive breast cancer. Breast cancer is the leading cause of cancer deaths in women, and breast cancer is classified by the expression of receptors, with estrogen receptor positive representing about 65% of all breast cancer cases. Currently, there are three main classes of standard therapies for ER-positive breast cancer. The first is the selective estrogen receptor modulators, or SERMs. The best known example in this class is tamoxifen. A second class is called the aromatase inhibitors, which block biosynthesis of estrogen, such as letrozole. A third class is represented by fulvestrant, which was initially discovered as a full antagonist and later found to also be a degrader of ER. However, fulvestrant is not orally bioavailable and is administered by intramuscular injection. Despite the initial effective of the therapies listed above, unfortunately 20 to 30% of patients eventually relapse and become drug resistant. A main mechanism of resistance was shown to be ERA mutations that allow ligand-independent constitutive activation of ER. The most prevalent mutations are Y537S and D538G. Therefore, more efficacious and safer therapies are still urgently needed. We were attracted to the dual mechanism of fulvestrant and our vision for a best-in-class molecule with the following attributes: a highly potent SERT and a full antagonist with the excellent potency maintained across ER-positive cell lines. We also desire a compound with excellent PK profiles, particularly with excellent oral exposure. But we also envision a best-in-class molecule with high safety margins and a low bill burden. To accurately measure the ER degradation in cells after compound treatment, scientists at Sargon developed a high-throughput in-cell Western assay. And the dose response curve of three compounds in this assay is shown to the left of this slide. And on the table to the right, two parameters can be derived from each compound. First, an IC50, which is the concentration at which compounds can achieve half its maximum degradation. And second, the degradation efficiency, the S-in value, which represents the maximum ER degradation that can be achieved at infinite compound concentration. The goal of Mack and team was to optimize both degradation potency as well as the degradation efficiency. In 2014, Genentech acquired Sargon and brought two clinical compounds into our portfolio. However, after profiling both compounds in our own assays, we recognized that there were risks for both molecules, as summarized by the table on this slide. GDC-810 showed excellent PK, but it was less potent in vitro, and it was a partial agonist on the uterus, while GDC-927 was more potent and a pure antagonist on uterus. It has PK risk with high clearance and poor oral bioavailability. At that time, we hypothesized that the partial agonist effect of GDC-810 was associated with the acrylic acid group. As a result, we decided to focus on the basic amino side chain of GDC-927 and try to improve its PK profile by switching to a different core. We quickly examined a diverse set of cores from previously published ER ligands and realized that we faced a dilemma. In most ER ligands, the phenol group, as highlighted in red, was required for potency, but the phenol was also the major site of oxidative and phase 2 metabolism. However, an exception was the tetrahydrocarbonyl or THC core present in AZD9496 reported by scientists at AstraZeneca. This core did not contain a phenol moiety, and we suspected that the absence of a phenol in THC core might provide an improved PK profiles and decided to investigate this core with a basic amino group, even though at that time we were not confident if such molecules would have good potency. To test our hypothesis, we prepared a couple of prototype molecules as represented by compound 1 and 2 and profiled them extensively alongside with AZD9496 and fulvestrant. In ER alpha degradation assay, it was interesting to note that even though all compounds show good degradation in MCF7 cells, AZD9496 showed a significant drop in degradation efficiency or the S-in value in T47D cells. To rule out that this was not due to an assay artifact, we conducted Western blot analysis in both cell lines and confirmed that indeed all compounds were efficient degrader of ERA in MCF7 cells. However, AZD9496 was less efficient degrader in T47D cells. The binding potency of the compounds with ER-alpha was further evaluated in PGC1A co-activator binding assay with either the wild type or Y537S or D538G mutants. As you can quickly see in this table, all three compounds show better potency with the wild type than with the two ER mutants. And it was very encouraging to see that compound 1 and 2 were more potent than AZD9496 against the wild type, Y537S, and D538G mutants, respectively. We believe the improved potency of the basic analogs relative to AZD9496 could be explained by its co-crystal structure of compound 2 with ER shown on the picture to the left, compared with the published co-crystal structure of AZD9496 shown on the right. The binding mode of the core with ER is almost identical in both molecules. However, for compound 2, the basic azetidine nitrogen was about 2.9 angstrom away from aspartate 351, suggesting a strong ionic interaction. This is in contrast to an unusual but still favorable acid-to-acid interaction between the acrylic acid of AZD9496 and aspartate 351. We went on to examine the metabolic stability of compound 1 and 2 relative to AZD9496 and GDC927. As you might recall, our initial hypothesis was that the THC core might potentially provide an advantage in terms of improved metabolic stability and oral exposure due to its lack of a phenol group. In vitro, compound 1 and 2 were found to be significantly more stable than GDC927 across species in both liver microsome and hepatocytes. We were encouraged to find that the improved in vitro metabolic stability also translated to better in vivo PK profile. As you can see in rat, dog, and cyno, compound 1 and 2 show lower clearance and better oral bioavailability than GDC927, confirming our initial hypothesis that the THC core might offer an opportunity for improved oral exposure. We also investigated the compound's effect on the uterus. The gold standard for evaluating uterotrophic effect in vivo is the uterus web weight or UWW assay. In this assay, agonist estradiol and partial agonist tamoxifen were included as positive control. From this assay, it was noteworthy that compound 2 showed no partial agonist effect, while AZD9496 was a partial agonist, although to a much smaller extent than tamoxifen. The uterus web weight data were further supported with uterus cell wall measurements for both compounds. In this picture, you can clearly see that the uterus cell wall after AZD9496 treatment was significantly thicker than that of compound 2. With all the data shown above, we concluded that the THC scaffold showed potential to realize our vision for a best-in-class molecule. We scrutinized compound 1 and 2 more critically against our vision for a best-in-class molecule. When compared to AZD9496 and GDC927 as shown in this table, we noted that there might be room to improve the potency of both compounds. For example, in the MCF7 anti-proliferation assay, neither compound was as potent as GDC927. Additionally, in terms of physiochemical properties, we were concerned that both compound 1 and 2 were quite lipophilic with high log D. As a result, both compounds showed poor kinetic as well as thermodynamic solubility. Therefore, we decided to further optimize the potency and physiochemical properties. Towards our goal of improving potency while reducing lipophilicity of THC analogs, we faced significant challenges as the ligand binding pocket of ER-alpha is quite greasy. Shown on this picture is the crystal structure of compound 1 with ER-alpha, and I color-coded the protein surface based on lipophilicity, with green representing nonpolar and purple indicating polar. As you can quickly see, the binding pocket is quite lipophilic and therefore prefers lipophilic ligands. This is consistent with the fact that historical SERMs or SERG showed high lipophilicity. The only polar interaction was the ionic interaction between the basic azetidine nitrogen with aspartate 351 and the hydrogen bonding interaction between the indole NH and the backbone carbonyl of leucine 346. Upon close inspection, we recognize that histidine 524 is about 4.4 angstrom away from the methyl group of the N-alkyl, and we wonder if we could introduce hydrogen bonding interaction with histidine 524 in order to fulfill our goal of achieving better potency while reducing lipophilicity. To test this hypothesis, we explored a wide variety of alkyl group at this position that could potentially achieve hydrogen bonding interaction with histidine 524. A breakthrough discovery was made when we found that the difluoroalcohol, as represented by compound 3, showed improved potency and reduced lipophilicity when compared to compound 1. We later confirmed that the difluoroalcohol was making a hydrogen bonding interaction with histidine 524 with a co-crystal structure with ER-alpha. As shown on this picture below is the co-crystal structure of compound 3 colored in sand overlaid with the co-crystal structure of compound 1 colored in blue. The hydroxyl group is about 2.8 angstrom away from histidine 524, clearly within the range of hydrogen bonding. We were quite excited about the discovery of difluoroalcohol and decided to combine it with O-azatidine amino group present in compound 2, which tended to give us better ER-alpha degradation. This led to compound 4, and its profile is shown on this slide. When compared to compound 3, it was gratifying to see slightly improved degradation potency and efficiency. However, somewhat unexpectedly, compound 4 showed surprisingly poor metabolic stability as measured in both liver microsome and hepatocytes, particularly with human. As a result, we couldn't progress this compound further in our assay cascade. Fortunately, while we were exploring various N-alkyl groups, we had a parallel effort that focused on examining various benzylic modifications. In this effort, we identified NH-azatidine as represented by compound 5. The profiles of compound 5 are shown on this slide. When compared to compound 2, compound 5 showed comparable antagonist and degradation potency and efficiency. We were pleased to observe that compound 5 seemed to be metabolically more stable, especially in hepatocytes, than compound 2. Additionally, the replacement of O-azatidine in compound 2 with NH-azatidine as in compound 5 also resulted in slightly lower lipophilicity. We attributed the lower lipophilicity of compound 5 to the more basic azatidine as shown in the structures, with a measured pKa of compound 5 about 1 log unit higher than that of O-azatidine. We were clearly aware that we had introduced an aniline into our compound 5. However, it was reassuring to note that no time-dependent CYP inhibition or GSH-ADA formation was observed for compound 5, suggesting that this aniline was not particularly reactive, maybe due to the two highly electron withdrawing fluorines on the phenyl ring to which the aniline was attached. As you might have guessed, subsequently we combined the difluoroalcohol and NH-azatidine, which led to GDC 9545, with its profile shown on this slide, along with compound 4 and 5. We were delighted to see that when comparing GDC 9545 with compound 5, the introduction of difluoroalcohol further improved antagonist potency, degradation potency, and efficiency, and resulted in lower lipophilicity with a measured log D of 2.48, as well as better kinetic solubility, consistent with what we had seen previously. In addition, replacement of O-azidine in compound 4 with NH-azidine gave GDC 9545 with improved metabolic stability when compared to compound 4. We further confirmed the binding mode and interactions of GDC 9545 with ER-alpha with a co-crystal structure as shown on this slide. Again, we saw the hydroxyl group is about 2.8 angstrom away from histidine 524, suggesting an hydrogen bonding interaction between the two, and an ionic interaction between the basic azidine nitrogen and aspartate 351. In the next 3 slides, I will share with you the novel discoveries on the mechanism of action studies. First, we asked whether every compound could degrade ER-alpha equally in a spectrum of ER-positive cell lines. Here is the Western blot data. In MCF7 and T47D cell lines on the top, almost all compounds promoted ER turnover to a varying degree compared to the DMSO control. However, this does not seem to be the case for the rest of 5 other cell lines for GDC-A10. In contrast, degradation of ER-alpha in all cell lines is efficient by fulvestrant, GDC-927, and GDC-9545. In addition, GDC-9545 is more efficient than GDC-927 in ER-alpha degradation across the board, and is even more efficient than fulvestrant in gamma-1 cell line at the bottom. Given the difference in ER-alpha degradation across the cell lines, we then look at the gene expression profile downstream of ER-alpha signaling through RNA-seq experiments. For simplicity, expression of one representative gene is shown on the y-axis. In MCF7 breast cancer cell line, 4-hydroxytamoxifen, a non-degrader, shown in black bar, showed partial agonism, while the rest of molecules, all degraders, exhibited no agonism. In HCC 1500 breast cancer cell line, however, both 4-hydroxytamoxifen and GDC-A10 did not degrade ER-alpha, and both exhibited partial agonism as shown by the black and salmon-colored bar pointing upwards. In other 3 compounds, including GDC-9545, are all degraders, and they suppress gene expression pointing downwards. Other than degradation and signaling activity in breast cancer cell lines I just shared, GDC-A10 and GDC-9545 also differ in 3 other areas, such as signaling activity in uterus, coactivator binding, and ER-alpha mobility. These observations were published in Cell last year as shown at the bottom of this slide. The take-home message is that we categorize GDC-9545 and GDC-A10 as 2 different therapeutic classes mechanistically, namely GDC-9545 as a full antagonist and SERD, and GDC-A10 as SERM-SERD hybrid. Our hypothesis is that complete shutdown of pathway will lead to superior anti-proliferative activity. As shown here, we profile compounds in a spectrum of cell assays including antagonism, degradation, and anti-proliferation. We also look at the compounds against both wild type as well as Y537S mutants. From these slides, it's clear that GDC-927 and GDC-9545 are superior to GDC-A10 in all cell potencies by huge margins, from 30 up to 100-fold. In addition, GDC-9545 is more efficient in degrading both wild-type and Y537S mutants than GDC-927 as representative by the SE number or the maximum degradation values. This slide shows that GDC-9545 not only compares favorably to GDC-927 and GDC-A10, but also to the historical SERMs and the more recently reported SERDs, such as Sanofi's SAR 439-859, Radius 1901, and H3B 6545. This is the so-called lipophilicity efficiency plot, in which the x-axis is log D, the compound's lipophilicity, and the y-axis being the PIC50 of anti-proliferation assay, with LLE equals 4, 5, 6, and 7 shown. Obviously, the higher the LLE, the more drug-like a compound is. GDC 9545 showed LLE of 6.4, the highest among all SERMs and SERTs that went into the clinic. This graph also highlighted our SAR progression, starting with compound 2, introducing the difluoroalcohol followed by replacement of O-azidine with NH-azidine. Two modifications that each increased compound's potency while reducing lipophilicity eventually led to GDC 9545. Since GDC 9545 and GDC 927 are similar mechanistically, we compare their DMPK and physicochemical properties as shown on this slide. GDC 9545 in green bars had lower clearance on the left and higher oral bioavailability on the right than GDC 927 almost across all preclinical species. In addition, GDC 9545 showed better solubility than GDC 927 as shown by both the kinetic and thermodynamic solubility. Here is the tumor efficacy data for GDC 9545 with the patient-derived xenograft model HCI013 harboring the Y537S ER-alpha mutation that drives the resistance to aromatase inhibitors. Fulvestrant and GDC-810 were both active in this model by slowing down tumor growth. GDC-927 did better when tested in this model, resulting in a really nice tumor regression, albeit at a high dose of 100 mg per kg. What is remarkable about GDC 9545 shown to the right is that it can achieve similar efficacy as GDC 927, but at a 100-fold lower dose. Based on the efficacy data, GDC 9545 is predicted to be a low-dose drug around 1 mg QD as a human efficacious dose targeting the maximum tumor growth inhibition in mouse, significantly lower than human dose projection for GDC 927 and GDC 810, respectively. In pilot tox studies, such as rat 7-day study shown on this slide, GDC 9545 in green dots showed orders of magnitude higher therapeutic index than GDC 927 in yellow and GDC 810 in red. All three compounds can achieve similar plasma exposure as shown on the x-axis. It was remarkable to see that GDC 9545 showed greater than 1,000-fold safety margin in all three doses. In addition, GDC 9545 lacked mortality or GI findings such as watery feces and dehydration, which were observed in both GDC 927 and GDC 810 studies. Sino's 7-day pilot tox show similar trends. This collection of desirable properties placed GDC 9545 in a rather unique situation. That is, the projected efficacious dose, represented by the blue dot at the bottom, is 100-fold lower than the tox-enabled starting dose, represented by the pink triangle on the top. It is usually the opposite for oncology drugs, in that tox-enabled starting dose is below projected efficacious dose. What this unique scenario offered us was the opportunity to choose a phase 1 starting dose in between, already efficacious while can further escalate up without significant safety concerns. After extensive preclinical work, it was really gratifying to finally see the drug concentrations of GDC 9545 when dosed orally QD in human, as shown in the graph on this slide. It is evident that GDC 9545 showed excellent pharmacokinetic properties in human. The 30 mg and 90 mg cohorts of GDC 9545 exhibited higher plasma level than either GDC 927 or fulvestrant at their clinically relevant doses. In addition, GDC 9545 exposure increased dose proportionally as shown on the table to the right. The Cmax to Cmin ratio was low, roughly twofold, minimizing any Cmax driven off-target toxicity. Terminal half-life of GDC 9545 is about 30 to 40 hours, supporting once daily oral dosing. Bottom line is that GDC 9545 has a good chance to achieve maximum efficacy in patients with excellent potency and high oral exposure. In summary, we demonstrated that GDC 9545 is a best-in-class molecule preclinically in all aspects, including its mechanism of action, potency, pharmacokinetics, efficacy, and safety. Currently, GDC 9545 is in phase 1 clinical studies. It is well tolerated with PK supporting once daily dosing. Robust target engagement and encouraging anti-tumor activity has been observed in heavily pretreated patients. Phase 2 and 3 trials for GDC 9545 are being planned. With that, I would like to acknowledge the phenomenal project teams at both research and development within Genentech. It really requires close collaboration and hard work by so many talented individuals to arrive at where we are today. Last but not the least, I also want to thank all the patients and clinical investigators for participating in our trials. Thank you for your attention, and I will be happy to answer your questions.